has been exposed to DNA that contains some molecule. In this case, it contains horseradish peroxidase, but that's not important. It could contain all kinds of different things, even a fluorophore. So it could be fluorescent. So that those small pieces of DNA are bound to the RNA molecules that are in this tissue, and a subsequent chemical reaction turns that black. So all of a sudden, we can look at an organism, and we can say instantaneously, my gene is at least being transcribed in this region right here. So we can, we're going sort of then from the bioinformatics to some of the functionality of that gene product. What's it doing? How are we? Where is it? And that sort of thing. And so here's just this is just a sort of another picture. We're not really. This was just looking at the DNA that's in here. So you've got a whole bunch of nuclei. This one right here doesn't express this transcript. We know that already. And when it's treated, you don't see any darkening. So we can tell very specifically our RNA is being produced right here. So it's kind of a cool way of looking at expression. Here is an example of. Would somebody turn the lights off over there just for a second? Thank you. You should give me the light switch up here too. So it's kind of hard to see, but here's an example of a worm. This is an example where the reporters are. This one has two reporters in it. So this is a reporter, and one is GFP. Green fluorescent protein. So it comes from a gene that was discovered in jellyfish, and it encodes a protein that is green under when light is shined on it. Typically blue light, it shines green, and it's non-toxic to an organism. And so you can get pets now. I think you can get mice or dogs. I can't remember. There's something. Is it cats?
They made dogs in Korea, and they make fish here, but you can't buy them in California.
Yeah. So you can get organisms that express this, and you put them under a black light, and they're green, and it's pretty cool.
That's awesome.
So this one also has another one called M. cherry, which is another molecule that is red under typically green light. So you shine green light, and it's very difficult to see, but there's some red up here in the mouth parts of the worm, and then both green and red are expressed pretty equally along the gut. Now this one right here is one that we call. So let me erase this, and I'm going to talk about reporters right now. Kind of two types. There's two types of reporters. One is what we call transcriptional. And this is where we simply replace the coding sequence. So I'll abbreviate that CDS, right? We did that before. Abbreviated coding sequence, CDS. So we replace the coding sequence of our gene with, say, GFP. Okay? So our original gene sequence is gone. We have the upstream sequence, the promoter, the downstream sequence, the 3' UTR, but that's all surrounding just GFP. So what that does is it just tells us the tissues that our gene is expressed in. It tells us where and when, what cell types, and when that gene is being expressed. But the GFP is just going to fill each cell. Yeah?
Why didn't you replace it? Why don't you just put it with it? What's the point of that?
That's a great question. Why don't you just put it with it? So it turns out if you just replace the coding sequence with GFP, it works almost every time. And everybody's happy. And, you know, it's time to celebrate. And the whole cell floods with GFP. So it's incredibly bright, and it's easy to see. The other type of reporter, can I erase this now? Everybody got this down? Is the kind you're talking about. And that's what we call a translational reporter. And that's when what we want to do is fuse the original coding sequence with GFP or mcherry or one of these things. Can you guys see when I'm writing up here even though the lights are off? Okay. So this is one like you're talking about. So what we want is far and away is better than the transcriptional, but they don't always work. Sometimes the GFP will cause problems for our original protein, and it won't work properly, it won't target properly, it'll get degraded in the cell, all kinds of problems can happen. So the transcriptional tends to work all the time. The translational, yeah, it doesn't always work. Yeah?
What is preventing the this transcriptional GFP just plain killing the cells and it just lost the important protein?
That's a great question, and the answer to that is that the organism still has the original gene. You're putting it in excess, so you're putting in extra copies that are like this. You're not wiping out the original gene. You're putting in an extra copy. Okay, so it still has its original protein. Good question. Okay, so let's take a look at some other transcriptional and translational. So this is a transcriptional fusion, a gene called Sp46, and here you can see it filling cells in a gonad, various places.